The events between fertilization and implantation comprise early embryonic development. In this video, I am going to tell you what are these events. Hey everyone, welcome back to hmlearnings. I am Harshita, the creator of hmlearnings, where students come to clear their concept and to get the study material. Make sure you subscribe my channel. So today we are back with the next video on the reproductive physiology, that is the early embryonic development. So in the previous video we have talked about the fertilization, and now we will see that what are the events which are happening between fertilization and the implantation. So usually it takes 6 days from the fertilization till the time the implantation of the embryo starts. So in that 6 days, what are the events which are going to happen, and that 6 days are called as the first week of embryonic development. So we know that the site of the implantation is uterus, and we know that the site of the fertilization was the fallopian tube or the ampulla region of the fallopian tube. So now the conceptus or zygote or embryo is going to be transported from the site of fertilization to the site of implantation by the ciliary beating and the smooth muscle contraction of the fallopian tube. Then it takes 6 days from fertilization till the time the first stage of implantation will start, and that 6 days is being called as the first week of embryonic development. And during these 6 days, the events which are happening include cleavage, blastulation, and the hatching of blastocyst from the zona pellucida. So in this video we are going to look in these events in detail. So as I said that this is a diagram of a female reproductive system. So you can see here that the fertilization has happened in the ampulla region of the fallopian tube, and if you remember that fertilization ultimately resulted into the first meiotic cleavage and the formation of the second cell stage. So the second cell stage has been formed here, and now from second cell stage it will ultimately, through a series of meiotic divisions, it will finally form a 16 cell stage called as a morula. Then it will enter into the uterus, and then it will adhere to the uterine wall. So if I say that during 14th day the ovulation has happened, during 16th day the fertilization has happened, and it takes 6 days from here, from the ampulla region to the site of the implantation, it takes 6 days to finally start the implantation. So how does the 3 events, the cleavage, blastulation, and the hatching is going to happen? So after fertilization or soon after fertilization, the cleavage is going to start. That is from the 16th day only the cleavage will start. The first meiotic cleavage will start. Then it will continue for about 3 days. Till 18th day the cleavage will happen, and all these events are been happening in the fallopian tube. So you will see that from the second cell stage till it forms a 16th cell stage called as a morula, it is happening in the fallopian tube. Now this morula is going to enter into the uterine cavity, but it is not being attached to the uterine cavity, it is freely lying in the uterine cavity. So now from 19th to 20th, the process which is going to happen is a blastulation, that is the formation of a blastula or the formation of a blastocyst. So formation of a blastocyst is going to happen, called as in blastulation. It takes about 2 days, and you know that now the conceptus has finally entered into the uterus. Now in the remaining 2 days, what is going to happen? The hatching is going to happen, in which the zona pellucida is going to be degraded, and now the conceptus or the embryo is going to start opposing to the uterine cavity, which is the first step of the implantation. So this whole is comprising the first week of the embryonic development. So if I draw this, so it will take from 16th day to 22nd day, that is the 6 days, and forming the first week of embryonic development. So we will see first cleavage. What is cleavage? So cleavage is defined as a rapid it's basically a series of rapid meiotic divisions that immediately follow the fertilization. So soon after the fertilization, what happens that the zygote will undergo into the meiotic cleavages, and it will form a bunch of cells which are called as a blastomeres. So during cleavage, what happens that the enormous volume of zygote cytoplasm is going to be divided into the smaller cells called as a blastomeres. So you can see here, what does it mean that if you see in this diagram that the size of this cell or the size of the embryo is the same only, and inside you can see that the number of the cells are increasing as the cell is undergoing the repeated mitotic divisions, but the size of the cell is reducing. So the number is increasing but the size of the cell is reducing. So if you know that during cell cycle what happens, that there is increase in the cytoplasm, then there is DNA replication, then it will be followed by the division of a cell into the two daughter cells. But here there is no increase in the cytoplasm of the cell. Whatever the cytoplasm the cell is already having, it is directly undergoing into the DNA replication followed by the meiotic division. So that is why this statement has been written that the enormous volume of zygote cytoplasm is going to be divided into the blastomeres. So all these cells are called as the blastomeres. So this is a timeline which we have already seen. So there are total 4 cleavages which are happening. So the first and the second cleavage is happening, they are very slow, and they are and it takes 2 days to complete the first 2 cleavages, and the third and the fourth cleavage is very rapid and it takes only 1 day to complete. So we will see that what it looks like, the first, second and third cleavage. So in the video of the fertilization, we have seen that fertilization ultimately lead to the first cleavage, and this first cleavage is forming the 2-cell stage. So you can see there are 2 cells here. Then there will be the third cleavage second cleavage, in which from 2-cell stage it will form the 4-cell stage. So you can see 4 cells here. Then it will undergo into the third cleavage, which is a comparatively rapid cleavage, and it will form a 8-cell stage. Then it will undergo a process called as a compaction that will be discussed in the next slide. And then after compaction, it will undergo into the fourth cleavage, which is ultimately leading to a 16-cell stage called as the morula. Okay, so with every cleavage there is increase in the number of the cells by 2. So first cleavage 2-cell stage, second cleavage 4-cell stage, third cleavage 8-cell stage, and then fourth cleavage a 16-cell stage called as a morula. So this happened in the fallopian tube. So now we will see the process called as a compaction which is happening between the third and the fourth cleavage. So what is compaction? So in this, this is a diagram you can see. So this is basically a 8-cell stage. So you can see here that you can count the 8 cells because their margins are sharp. They are well separated from each other. So they are loosely arranged in the early 8-cell stage. But in the 8-cell stage, what happens? This blastomeres will start expressing some cell adhesion molecules such as E-catherine, and this will maximize their cell-to-cell contact, and ultimately their margins will be decreased, and they will mixed with each other, form a compacted mass of the cells, and this is called as a compaction. So in the previous slide also you can see that here what happens that this is also 8-cell stage and this is also a 8-cell stage. So the difference between these two is that you can count here the 8 cells separately, but you can't say that there are 8 different cells here because of the process called as a compaction. They come in the close contact, they start having the tight junctions between them, they start having the gap junctions between them, and that is how they form a compacted ball of the cells. Okay, so they are having the tight junctions and they are having the gap junctions also. So the compacted 8 cell then undergo the 4th cleavage and form a 16-cell stage called as a morula. So morula means mulberry like. So this is the 16-cell stage which it forms. So now we have finished with the cleavage. The next event which is happening is the blastulation. So blastulation happens, it takes usually 1 to 2 days, and now your morula has entered into the uterus and it is freely lying in the uterine cavity. So morula is, if you see the morula diagram here, so it is having the external layer of the cells. This is the external layer of the cells, your external group of the cells, and this is the internal group of the cells. So now you see here that it is having the internal cells which are surrounded by the external cells. Okay, then the uterine fluid is going to enter inside the zona pellucida and it is going to create a fluid filled cavity called as a blastocyst. So if you see this diagram, so earlier you have seen that there was a ball of cells in which is a compacted thing. Okay, so there were so many cells which are compactively arranged with each other, but now there is a cavity which is being created because as the uterine fluid enters inside the conceptors and form a cavity called as a blastocyst. So this external layer has formed, is surrounded by us cells, and this is going to form a ring overall covering all the margins. This is forming the trophoblast, and the another part is the inner cell mass, which is some cells, the internal group of the cells are being pushed to the one side of the embryo, and this is going to form the inner cell mass. So this is the first differentiation step of the embryonic development, like which cell is going to form the trophoblast and which cell is going to form the inner cell mass. So some genes are playing the role that if these genes are being expressed by this cell, they are going to form the inner cell mass, and similarly for the trophoblast. So the trophoblast cells are the cells which is going to form all the extra embryonic tissue such as chorion and the placenta, and the inner cell mass is going to form the embryonic tissues. So we have seen now the cleavage and the blastulation. The last step is the hatching, which is happening again in 1 day or 2 days, followed by implantation. So now you have seen in the diagram that in all these 6 days or in the 5 days, initial 5 days of the embryonic development, early week of embryonic development, the conceptus or the embryo is being covered by the zona pellucida, and it is preventing its attachment to the fallopian tube or to the any other part which is not for the site of the implantation. So it is preventing that, but by day 22nd, the conceptus has entered in the uterus and it is near the site of the implantation of the uterus. So the zona pellucida will start degenerating and blastocyst will come in the contact with the uterine wall. So how does this happen? First thing is that there are some lytic factors which is released by the uterus itself, that could be a plasminogen. Then this is an inactive form. So the trophoblast is going to release a protease which will convert this plasminogen into the plasmin. Now this plasmin is going to cause the degradation of the zona pellucida, and hence there will be hatching of the blastocyst. So now the blastocyst is in a state to adhere to the uterine wall and beginning the process of implantation. So this is about this video of first week of embryonic development. These are the references you can look up to, and in the next video we are going to talk about the implantation. So if you like this video, please like, share and subscribe my channel. Till then, keep learning.